All right, welcome. Today I'm going to talk about how to read and interpret a cancer clinical trial. We're trying a new format here. We're doing this live in front of a real studio audience. No, in front of our research team. And these slides have been put together by Dr. Timothy Olivier, and I've actually never seen them before this moment. No, they're based on a post I had on the drug development letter, which was about how to read and interpret a clinical trial. We got Timothy here. We've got some other people here, and we're going to get into it. We're going to hopefully do some questions, and this should be an interesting framework to you all. So number one, when I read and interpret a cancer clinical trial, I always enter with 10 basic questions. 10 questions that I think frame the way you should think about the study, but then I think ultimately it's always going to have to come down to your own judgment and your own clinical acumen. So step number one, does the trial have a control arm? I think we take for granted in medicine that clinical studies are randomized, that they have control groups. That's not always the case. In oncology, in fact, about 1/3 of FDA drug approvals are on the basis of an uncontrolled study, meaning that they just gave a drug to 100 consecutive people, 50 consecutive people, and they saw what happened to those people. One measure that we commonly use in oncology is called a tumor response. It means that a person's baseline tumor has decreased in size by 30% or more. That's the RECIST 1.0 tumor response criteria. And the percent of people, of 100 people who get a cancer drug, whose tumor shrinks 30% or more, that's called the response rate. So here in a paper by Emerson Chen and myself, we looked at all of the drugs that were FDA approved on the basis of response rate without a control arm, and we plotted out what was the response rate for approval. And we put them in a few buckets. Those that got regular approval, meaning they had no post-marketing commitments, those that got accelerated approval, which was later converted, and those that got accelerated approval that was never converted. And basically what you find is that the median response rate is 41%, meaning that among the drugs approved based on response rate, no tumor, no control arm, they shrank the tumor 30% or more in 40% of the people. So that's what you're seeing in this figure here. Some drugs are much, much better, like Gleevec is a 100% response rate, but many drugs are much, much worse, and you get see like 16% for decitabine and enasidenib at 23%. Okay, the reason it's important to know if a trial has a control arm is that if it doesn't have a control arm, we tend to make historical comparisons. We tend to compare it to other benchmarks. These comparisons can be very misleading. This is a paper that Alison Haslam and myself did, where basically what we looked at was among drugs approved on the basis of response rate, we asked what was the response rate for a pre-existing therapy used in that space. And what's shown here is that the orange bars are the novel drug being approved based on response. The blue bars are the novel drug, and the orange bars are the precedent drug, the drug that was already there. And the point here is that the bars are roughly comparable. So if the FDA is saying that we're approving the new drug because you've never seen a response rate so good, well, that's not true. Often they have seen a response rate at least as good or better. And if you have two drugs with a similar response rate, then I do think you have equipoise and you could have run a randomized study instead. So I think this really calls into question the FDA's drug approval. But you should know that when you make historical controlled comparisons, those tend to exaggerate treatment efficacy. In sort of a very famous analysis of studies that were both studied in a historical control fashion and a randomized fashion, 70% of the historical controlled studies found a benefit. Well, that was only validated in 20% of the randomized studies. That's an old paper by Sachs and colleagues. Okay, so now let's say you're in the 2/3 of oncology studies that are randomized. Well, what should you be looking for now? Well, I think the first thing you should look for is the control arm. I always say a randomized trial can only change your practice if the control arm is as good as your practice. And here, this is an analysis by Talal Halal, where we looked at the quality of control arm for US FDA anti-cancer drug approvals, and we found rather clearly that 17% of US FDA randomized studies use a control arm that was suboptimal. And it's not suboptimal when the trial is being run. It's not suboptimal at the end of the study. It was suboptimal before the trial even accrued patients. And this to me is a huge problem of FDA leadership and accountability, where they're allowing people to run clinical trials against chlorambucil over and over again. Nivo versus dacarbazine in melanoma, an unethical control arm when you already have ipilimumab. And when you run trials with strawman control arms, olaparib versus giving a Enza or Abi to somebody who already had Enza or Abi, when you run these unethical control arms, I think you can't conclude that the novel drug is better than what doctors would actually do in practice, because your control arm is not what doctors are actually doing in practice. Another example led by Timothy, he took a look at when they do a randomized study against an investigator choice. The investigator is saying, hey, it's going to be the novel drug versus investigator choice. What could be better than that? We're going to show our new drug is better than what you would choose otherwise. But what he shows here is that the choice is not an unfettered choice. It's a restricted choice. So shown here in the dark bar are the restricted choices. Shown in the orange bar are the real choices, unlimited choices. What do I mean here? Basically, if you test the new drug against a limited selection of choices where you only pick the weakest options, it's easier to show that the new drug is a winner. I think the classic example that came out most recently is CAR T. CAR T is going up against, investigator choice chemotherapy, but you're not allowed to give Dara-KD. You're not allowed to give carfilzomib Pomdex. The most potent regimens you're not allowed to give, Isa-KD. If you're banning the most potent regimens, then it's a lot easier for CAR T to eke out the PFS win. So now let's say the control arm is something that satisfies you. Well, then the next question to ask is if the trial is adequately powered. Now, I think that people forget that an underpowered study actually suffers from two types of errors. So an underpowered study means that, the sample size is inadequate to make a firm assessment on the endpoint that you're talking about. It didn't have enough power to make an assessment there. And we tend to think of the errors that are being made in an underpowered study, which is that you fail to detect a difference when a difference actually exists. And that is, in fact, an actual error that occurs with underpowered studies. But what people forget is an underpowered study also means that when you do detect a difference, and it is statistically significant, it's much more likely to be exaggerated or spurious. And that's the part that people miss. So for instance, in BR plus or minus POLA, that is a study that was not powered for overall survival, and I would not trust that overall survival result, which is a huge overall survival victory and won't be replicated, and wasn't replicated, I think, in the frontline study. The other classic example is olartumab, which is Lartruvo, a soft tissue sarcoma drug, which in an underpowered phase 2 study found a very large PFS and OS benefit that was absolutely, totally null, superimposable Kaplan-Meier curves in the randomized study. So be careful about underpowered studies. If the study's underpowered, I think you can really only look at the primary endpoint of the study, a phase 2 study. You can't look at secondary endpoints. This also applies to oligometastatic randomized controlled trials. Was crossover done correctly? So I think crossover in oncology refers to unidirectional crossover, where if you were initially assigned the control arm regimen, you're allowed access to the experimental arm regimen, where if you were initially randomized to the experimental arm regimen, you were given standard of care therapy thereafter. And the reason people misunderstand crossover is that sometimes they have it and sometimes they don't have it, but what they forget is sometimes you want it, sometimes you don't want it, sometimes you get it, sometimes you don't get it, and there's actually 4 situations that may arise. So we frame this in our paper, when is crossover desirable and when is it problematic? All right. So crossover, so this is the 2x2 table. Crossover is either desirable or it's undesirable. It either occurred or it didn't occur. So it's desirable, I think, in situations where a drug has already shown a benefit in a latter line of therapy and you're moving it up front. And it's problematic in situations where a drug has never established fundamental efficacy. If it's never established fundamental efficacy, you don't want crossover. If it's already shown a benefit in the second line, you're trying to move it up, you do want everyone to get in the second line because you're asking the question, is it better to give it routinely up front versus giving it in the second line? And crossover can either occur or not occur. I'm not going to use the examples on this paper. I'm going to give some different examples. I think when we talk about avelumab maintenance in bladder cancer, the real question is, did the control arm get pembrolizumab at second line for urothelial cancer as they ought to have? And the answer is that they didn't. When we talk about pembroaxitinib versus sunitinib in RCC, the real question is, among people who progressed on sunitinib, did they get PD-1 therapy like they ought to have gotten? And the answer is they didn't. So how do I know pembroaxitinib is better than sunitinib followed by pembro? I don't know that, particularly for people with slowly progressive disease. Crossover is problematic, I think, in situations like the IDH inhibitor in glioblastoma. What's the name of that drug, Timothy? I'm forgetting that. Vorasidenib?
Yeah.
Vorasidenib. In the IDH inhibitor study, Indigo, they mandate crossover, even though vorasidenib has never established benefit in glioblastoma. in low-grade glioma. And so there is no OS benefit, but we don't know if that drug would have improved survival without crossover, or if in fact the harms of the therapy are actually falling on both arms and actually being washed away. And it actually would have reduced overall survival, but you don't see that because everyone ends up on the study drug. So was it done correctly? I always say that some people say that, look, if you randomize to an experimental arm and control arm and there is no overall survival benefit, that means that we should give the new drug earlier, that benefit was washed away with subsequent therapy. But the point I always make is that if at the end of the day, if the new drug can't eke out a survival benefit after many lines of therapy, then why do you even use the new drug? I mean, all you care about is the end of the marathon. This is a nice figure. And what? This is French.
So this was an illustration of this work we did together with Alison and you. And so basically we studied 3 years of FDA-approved drugs and 3 years of publication. And we find that maybe you can just click on the next slide, but the results were really sobering. First, it was not appropriately reported in many cases. And secondly, even when it was reported, we assessed, the quality of post-progression treatment, and we find it was optimal and appropriately reported in 12% of FDA-approved drugs. So this is just the illustration, the quantification of what you described before. Yeah.
Which begs the question, which is that a drug that has a benefit when all of the subsequent therapy is not up to the global US or Switzerland standard, what does that tell you about whether or not you should use it in the US or Switzerland? You want to know it has a benefit in the backdrop of the care you're providing in the US or Switzerland. And that's not the case in majority of these studies. I mean, one, they're not even telling you what happened post-progression, which deprives you of the ability to know. And then when they do tell you, it's mostly substandard. It's mostly beneath the global best standard of care. This happens because the companies go globally to run these studies in places that can't afford the subsequent therapy. But think about that. That study informs nobody. The country that they're running the trial in, they're not getting any benefit because when the trial's over, they can't afford the new drug. After all, they couldn't afford it anyway on the back end. They're not going to be able to afford it on the front end. And this information doesn't inform places like the US and Switzerland because we don't know if the new drug would have had a benefit against the backdrop of therapies we're giving. All right, so what's the endpoint of the study? This is the fifth point I like to think about. So I like the control arm, post-protocol. Is the endpoint something that's a surrogate endpoint, meaning that it's an endpoint the patient didn't know mattered until the doctor told them that it mattered? Or is it a clinical endpoint, living longer, living better, overall survival, health-related quality of life? And if it's a surrogate endpoint, does it have a good correlation with overall survival? So people forget that what is progression-free survival? Progression-free survival is the time until one of four things happen. Shown here initially is like the initial size of the tumor with a certain diameter. And progression-free survival is the time until one of four things happen. One, the patient could die. Two, there could be a new lesion on the CT scan that didn't exist prior, so the lungs were clear and we have new lesions. Three, the tumor could get bigger, and if it gets more than 20% bigger from the starting size, that's deemed progression. And then the fourth thing that could happen is the tumor initially shrinks. If it shrinks more than 30%, that's the response. And here we measure progression from the smallest it ever gets, the nadir value. And progression-free survival is a time to event, composite endpoint, meaning whichever one of these four things happen first is coded as the endpoint. Does it have a good correlation with survival? And the answer is, in an analysis done by Alison, that the strength of these correlations is quite poor. That across the metastatic setting, drugs that improve progression-free survival do not always, in fact seldom, and they have a poor correlation with drugs that later improve overall survival. Most of these correlations are low. There are few medium and there are few high. In select settings, PFS does predict overall survival, but that certainly is not the norm in oncology. And it's important, I think, to go to this paper and pull up a table of the correlation coefficients. Any other thoughts, Timothy?
Yeah, maybe the only thoughts I would have is the nice paper you did with Robert Kemp, that the correlation, the trial level correlation has to be done with a certain drug, certain setting, and in a sense, you should not rely on them because the landscape of treatment is moving so fast. I think we should really think about that and try to really keep clinical hard endpoint like overall survival and quality of life. This is a very good point you made with Robert Kemp in this paper.
Yeah, ultimately, patients care most about how long they live and how well they live. That's what they care about. And these are all just convenient ways we measure things that state investigators that are supposed to predict whether or not they live longer, live better. And along the lines of what you said is often the FDA produces analysis saying the FDA looked at all of the trials submitted to the FDA to ask whether or not MRD negativity predicts PFS in myeloma. Well, one of the things that they're not looking at is all of the trials not submitted to the FDA.
Not submitted, not published.
Not submitted. They're not looking at the lion's share of the data for most clinical questions. So when the FDA has a surrogacy analysis for trials submitted to the FDA, I usually take that with a grain of salt. They are missing the vast amount of data not submitted to the FDA. And of course, what's not submitted to the FDA? Negative data, data that's unflattering, data that doesn't fit our preconceived notions, data not used for drug approval. So their analyses, I think, are very low credibility, in my opinion. Quality of life. Now, we talked about survival, we talked about surrogates for survival, but what about quality of life? Quality of life is arguably more important to patients. I think if you survey people, they say they would rather have a drug that made them feel better than made them live longer. But the question is, when clinical studies measure quality of life, do they measure it properly? And in this paper led by Allison, what we looked at was, again, 2 bars here. The long bar is showing you how long people live with these different cancer conditions in the clinical study. And the short bar is showing you how long the quality of life forms were collected. And the point is that the short bar is much shorter than the long bar, meaning that we're collecting quality of life not for the patient's journey through cancer, but just for that initial period after starting the new drug. So when somebody tells me, hey, the drug has a health-related quality of life benefit, that doesn't mean you live longer for your entire cancer journey. You might just live better for the first 4 months, and then the aftermath might be horrific, living with toxicity, living with pain, and that's not even being captured in the forms. So this to me is such a failure of the way we collect quality of life. It's not even captured correctly. And it wouldn't cost them that much to do it correctly. Now, this is something that Timothy did, and maybe I'll walk you through it, and then let's see what Timothy has to say. This is a classic study that compared the combination of Nivo plus ipilimumab versus Nivo, followed by ipilimumab. I'm going to omit the ipilimumab-only arm for our discussion. We're really looking at the green curve and the blue curve. And in Checkmate 067, people say the PFS is better from Nivo-Ipi. Even though there's no OS benefit, we ought to give Nivo-Ipi, and in part because the quality of life with Nivo-Ipi is no worse than Nivo alone. But when you tell that to a doctor who gives Nivo-Ipi, they're going to say, that doesn't sound right. Nivo-Ipi is incredibly toxic. It blows out pituitaries, causes autoimmune hypophysitis. It causes all this IRAEs, immune-related adverse events. It's way worse than Nivo, but why is the quality of life no worse? And what Timothy investigated was that the grade 3-4 toxic events are way worse, but the quality of life is no worse. Why? And so what he shows in this figure and this next figure is basically the reason the quality of life is no worse is that many, many people are not completing the Nivo-Ipi form. Look at this. At time point 1, 237 people are completing the Nivo form for quality of life. Only 182 people are completing the Nivo-Ipi form. There's so much more missing data with Nivo-Ipi. Why is that? Why are some patients on Nivo-Ipi not having the strength to complete the form? And the answer is probably they're not feeling so good. In other words, they're only looking at the outcome of the form among people who complete the form, not people who don't complete the form. And the people who don't complete the form are probably not doing as good as the people who complete the form. So all of the averages they're getting are just among the completers and not the non-completion forms. And if you factored those in, you'd probably get way worse outcomes, which is in fact what Timothy does. He imputes different values. He shows that making modestly different assumptions about what happens to people who don't fill the form, this low-value to quality of life imputation method, Nivo plus Ipi, you can easily demonstrate that Nivo-Ipi is probably way worse in terms of quality of life than Nivo alone. Timothy, any comments on this paper of yours?
No, I think you explained it very clearly. I think you have always to think about your gut feeling. I mean, this paper was really driven by my gut feeling. When you see such differences in toxicities, and as you explained it, there can be long-lasting toxicities, you have to really dig into the data to see if there's something happening.
Yeah, I think that's absolutely right. That if the clinical trial report doesn't fit your intuition, so why is the adverse events twice as common, but quality of life is no worse? Then you have to dig into the data. There's probably some explanation there. And here, I think the likely explanation is that, well, the people who are having the IREs are not filling out the form.
Yeah.
And this is called informative censoring, which we published a lot about and more to come. Question number 7. All right. Yeah, it's not over yet. Question 7. Question 7 is the drug dosing fair? And often the clinical trial has a fair starting drug dosing. I'm thinking of the non-inferiority study, lenvatinib versus sorafenib for hepatocellular carcinoma. It's fair for the company to come up with whatever starting dose they want for lenvatinib and sorafenib. But here's the problem. The dose reduction rules may not be fair. In other words, if you have any side effects from lenva and side effects from sorafenib, are you having the same dose reductions? And the answer is here. If you have side effects from sorafenib, you go from 100% dose to 50% to 25% to stopping based on side effects. If you have side effects from lenva, you go from 100% to 67% to 33% to 17%. In other words, with the same side effect, you continue to take a higher dose of the novel drug than the control arm. And what this means is, in many cancers, pushing the dose matters, getting it to the optimal dose, particularly for like promiscuous tyrosine kinase inhibitors. The dose does matter for creating sort of stasis in terms of cellular cancer growth. And yet here, at every step, you're getting an advantage to lenvatinib. So the question is lenvatinib a better or similar molecule? Or is lenvatinib achieving its outcome because it has a more favorable drug dosing? In other words, do I need to pay all that money for the lenva, or could I just be better about pushing my sorafenib dosing? And in fact, what this paper shows is that even with comparable dose reductions of the two products, they got a lot more cumulative dose of lenva, probably because the drug dosing schemes were unfair. So how often does this happen? Does it occur? This is the paper by Timothy, and I'll let him walk you through it. Timothy, so what did you find when you looked at all the clinical studies on this topic?
I mean, yeah, that was really fascinating. So we basically selected all the trials where we could compare such rules of dose reduction and supportive care rules. And in 55% of the case, those were favoring the experimental arm. So this was really astonishing, such difference was impressive. So the conclusion, if you just click on is in those cases, I mean, we can't really rely totally on the reported results, because maybe this was driven by the dose intensity.
I think that's absolutely right. You don't know it's a better molecule. You don't know it's just the dose that matters. And I think that's a problem if the new molecule is substantively more expensive than the older molecule, which is often the case in oncology. Okay, I'm going to skip that for now.
Yeah.
Question number 8, does the product have value? So now let's say the control arm is correct, the post-protocol care is correct, the power is adequate, the endpoint is a clinical endpoint, there's no games around dose modification. Well, now the question comes is, does it have value? In other words, what is the dollar per life year gained? Societies, they have fixed amount of money, and we have to choose what to pay for. We're going to pay for better nutrition in elementary schools, or we're going to pay for better prenatal care, we're going to pay for these cancer drugs. And so calculating the dollar per quality is very important. One thing I always say with dollar per quality is we have to be very careful we're calculating it for the real world benefit. This paper that Sham and I did many years ago, we called overall survival in cancer drug trials a surrogate endpoint for real world overall survival, because the clinical trial patients are so dissimilar from the real world patients that outcomes in the real world are often way lower, way worse than what you get in clinical trials. So if a clinical trial tells you it's $200,000 per quality, don't be surprised if in the real world it's $600,000 or $800,000 per quality, because you're not getting as many qualities from that drug because the benefits are much smaller. This is a really good example of secatuzumab govitecan, led by Timothy. Timothy, walk us through this figure where you talk about...
I mean, the figure, there are some issue with the design, but the thing is, even in the real life, the rules that will be applied, for instance, for dose reduction, won't be the same. So in the real life, we know that the patient won't be the same. The treatment will be free for the investigator. It was a restricted choice in the trial. And so the question is really whether the benefits seen in the trial will translate into real-life patients. And so I felt it was a good example. The other example you often talk about is sorafenib in real life. This paper by Stacy Duzetsina, which is very impressive. But I mean, the overall point is what you said, yeah.
So let's do this one. So yeah, there's a 6-month OS benefit from secatuzumab, which is a lot. I mean, I think people are going to say that's a really impressive benefit. But now let's say, well, what if secatuzumab govitecan and physician choice had the same dose reduction scheme and the same use of GCSF? All right, well, maybe the benefit will be a little bit smaller. What if the control arm wasn't a restricted choice, but the doctor could do anything they otherwise would want to do? Maybe it wouldn't be 6, it would get a little bit smaller. What if the trial didn't halt early? Halting early exaggerates treatment benefit. We'll get a little bit smaller. And what if it wasn't open label but double dummy or blinded design? it would get even smaller. And so these design features exaggerate that benefit. And then the last point is, what if it wasn't run on clinical trial participants, but run in a registry-based, pragmatic, randomized trial on average people? It would be even smaller, and all of these outcomes would get worse. And so what looks like a 6-month benefit in a clinical trial in real life might be, a 1-month benefit, a 2-month benefit, a 3-month benefit, or no benefit at all. We have no idea because we don't have a study that's done in the real-world population. And so what's the dollar per quality-adjusted life year here? I think it's probably horrendous. It's probably horrendous just based on the trial. It's even more horrendous if you factor in those biases. Last 2 points. If a trial is non-inferiority, a particular type of study design where you try to establish not that a new product is superior to an older product, but that a new product is no worse than some pre-specified margin or delta, then there are special questions to ask. One of those questions is, why are you running a non-inferiority study? To run a non-inferiority study, your new drug has to be either cheaper, more convenient, or less toxic than the old drug. When Allison and I looked at these studies, we found that many times the new drug is just as expensive, just as toxic, and just as inconvenient. In other words, why do you need to run a non-inferiority study if you have no other advantage? You should run a superiority study. The next point is, how big is the margin or delta? I always say, it's difficult to parallel park a car in a San Francisco street on a hill with, barely enough space to fit the car. But anyone can parallel park a car in Kansas in a parking spot the size of a school bus. In other words, with a very big or permissive margin, it's easier to parallel park. Similarly, if you pick non-inferiority margins that are so big, then everything will be non-inferior. Even placebo is non-inferior to active cancer drugs with a big enough non-inferiority margin. And most cancer clinical trials that are non-inferior are using horrendous margins that guarantee and ensure non-inferiority. Question 10. This is about developing judgment, and I'm going to have Timothy talk about it. But I think that this is the hardest thing to teach, which is that no matter what your checklist looks like for a clinical trial, it's never going to be complete. And you always have to have judgment, which is you have to really sort of understand the disease process, understand how you would treat it otherwise, and think about the fact that the person running the clinical study has a strong vested interest in getting a positive result. And they're often gaming the clinical study in a way that you do not see, you do not fully appreciate, in a way that may even be hidden and not captured by these rules, because there's many more ways to cheat than there are checklists to catch cheaters. And because the rabbit runs faster than the fox, because the rabbit runs for his life and the fox runs for a meal. In other words, that gaming a clinical study beyond available scores will always be the case because the person gaming it has an incentive to come up with the next game so that they can achieve a victory. Timothy, what are your thoughts on this? This is your 10th point.
I think the last point is really, really important. And I would say it's good to be prepared even before reading a trial. I mean, it's good to really approach a trial with which are your questions, what is my current practice, and then you will really be able to analyze the trial with this framework. If you just read it from cover to cover, I think you can really miss the point because you are not really knowing what you are looking for. So I think it's the work you are doing and we are doing here, try to really raise awareness about some of the limitations. And you're right. I mean, for instance, we didn't talk about informative censoring, which is very important. We didn't talk about many, many other topics, and the list could go on and on. But I think if you take the control arm, post-progression, crossover, all the points you made, it's already, a very, very good filter to approach trials.
Yeah, this should be called like How to Do Journal Club. The last thing I'd say is that for listeners who are interested, you can go check out Drug Development Letter right now. Develop drugs. What's the website, Timothy?
Drugdevletter.com.
Drugdevletter.com. You have forthcoming in ASCO is the LAURA trial. Yeah. And Timothy has what I'm going to look for in the LAURA trial. He's already anticipated what I think the limitations of that study are, and he's framed them in his discussion, which I think is great. So here are the 10 rules for Journal Club. Maybe we should title this when you post it called, How to Do a Great Cancer Journal Club. 1, is there a control arm? If so, is the control arm what you would do in your clinic otherwise? Is the trial have adequate power for the primary endpoint? Are there issues of crossover? What was the primary endpoint, clinical or surrogate? And what is the strength of the surrogacy? Is quality of life reliable? Were the drug dosing rules fair? What's the value, and what's that value in the real world? If it's non-inferiority, we got some special considerations. And finally, to practice and develop judgment. So I'm going to stop there, take questions from you all, and I hope this video is of interest to people.
Thank you.
So what questions you got?
I have a question right off the bat. So you
Hit it.
mentioned
Yeah.
you mentioned PFS, and you really showed that PFS doesn't really correlate with overall survival that well in most metastatic cancers. But I often hear that people say, oh, they patients feel better when they're told their disease hasn't progressed by their doctor. So is there any evidence that PFS correlates with quality of life?
Yes, good question. So there are two analyses to my knowledge, one by Kovacs and one by Gaywali, that look at the correlation between drugs that improve progression-free survival and whether or not those drugs improve health-related quality of life. Both of those studies find that there is poor correlation, that drugs that improve progression-free survival don't improve health-related quality of life. As a general rule, I mean, the correlation is very poor. But let me come to your point directly. I know an investigator who, in one clinical study, documented quality of life before and after the investigator told the patient, you have radiographic progression. So the investigator goes in, they do quality of life form, and then they say, oh, by the way, your tumor has progressed and you have to come off the study. They do the same form again. And the answer is the quality of life deteriorates rapidly. Telling somebody they progressed, even if it's arbitrary, changes their perception of their disease, right? Wouldn't it be terrible to be told you progress, even if it doesn't change how you feel? So two things are true. One is these arbitrary numerical cutoffs for radiographs don't predict when people start to feel crummy. They don't predict when you feel better or feel crummy. But telling somebody that it's getting worse, will make people feel crummy. If you actually walked into rooms and you were deceptive, which I don't encourage anyone to do, but you lied to people and said, your tumor's getting worse, or you lied to them and said, your tumor's shrinking, I don't see any evidence of it, people will say they feel better, right? So I think we have to be very careful to distinguish between quality of life effects on perception and quality of life effects that are, I think, real and enduring. And so I think that's why both things are true. Investigators feel like telling someone they progressed deteriorates quality of life. I think that's true, even if these arbitrary PFS cutoffs don't actually predict better quality of life. And then the final thing I say is, if people really think their drug improves quality of life, you can prove that. Just document that health-related quality of life is better. And do it correctly, measured in everybody, and collect it over time. The last thing I'd say, most of the drugs that improve quality of life, the vast majority, also improve overall survival. It's very difficult to make a drug in cancer that makes you really feel better that doesn't also extend your life. Are you satisfied?
Yeah, very satisfied. I think that makes total sense. And I think that's what I see in practice too. Like, anecdotally, obviously, if you tell someone that they're progressing, they're going to feel bad. Yeah. But that doesn't mean PFS correlates with quality of life or, the patient feeling better.
Yeah, I think that's the case. And I think a lot of people like PFS because they're saying, well, who would want your cancer to progress? I mean, that's true. Who would want your A1C to go up and who would want an LDL of 130? I mean, nobody wants these things, but we have to be careful. The reason you don't want an LDL of 130 is the doctor has inculcated you into believing LDL of 130 is bad, rather than the real reason you want an LDL of 130 is you have a higher risk of MI. And so we have to be careful. Is it the case that PFS means higher risk of MI, i.e. like you're less likely to live longer, or are you just chasing a number? Are you just chasing some arbitrary thing that doesn't predict how your life is? Any other questions? All right, I guess if not, we're going to stop. Go ahead, hit us.
So you mentioned how in clinical trials, the patients can often be much healthier than the patients you might see in the real world or in clinic day to day. Are there any good methods of capturing data after the clinical trial and following up patients maybe in the real world? And then if there are the results drastically different to the trials? Like how much difference do you think it makes the patient selection in the clinical trials as opposed to the patients that you might see more routinely in the real world?
Great question. So in the pivotal study, the SHARP study, randomized trial of placebo versus sorafenib, the control arm lived 8 months, the treatment arm lived 11 months. 8 to 11. When Stacy Ducet-Zena took the same drug, sorafenib, and looked at all the people in Medicare who got it in the years that followed, the Medicare beneficiaries, remember, placebo is 8, treatment is 11. The people in the real world who got the actual drug, they should be looking for 11, they actually got 4.5. People in the real world getting the active drug lived half as long as people on the clinical trial getting placebo. Okay, that's how bad it is. And that's just one example, but we've documented it in a paper with Sherry Aspinall and RCC. It's been documented in many, many studies. It's called the efficacy effectiveness gap. But the crux of the problem is that the real world clinical outcomes are so much worse than the patients on study. often half as bad, like 50%, that's not an unreasonable figure. And that's because on a study, there are implicit exclusion criteria, like your renal function has to be X, your bilirubin has to be Y, your Charles Pugh score has to be, good. But so those are the explicit inclusion criteria. There are also implicit inclusion criteria and criteria where nobody says it's an exclusion criteria, but if somebody has a poor social network or they just seem flaky, they seem like a flaky person, they're not going to come back. Are you really going to put them on a study? They're not going to come back. They're flaky. Well, flaky people might have worse disease. They might have tougher socioeconomic situations. They might have less family support. And those things probably factor into their clinical outcomes. So all of these eyeball tests that doctors use for trials, plus the actual stated criteria, result in very different groups of people being studied. And we have that paper, and I think in Nature Reviews Clinical Oncology, where we solve the problem, where we say that cancer trials need to be a lot more like the UK recovery study for COVID-19, where you just try to enroll almost everybody, a large pragmatic study. And then later you can ask if some subgroups derive benefit and other groups don't. Like people with good bilirubin, do they get the benefit, but not people with deranged bilirubin? But if you don't do those kind of big pragmatic studies, then you really never know about the average person in the country. To answer your question, yeah.
Yeah.
Any other thoughts? Timothy, closing thoughts?
On that positive note.
All right, we'll stop the video now, but hopefully this was of interest to people. We'll try to post this on our drug development letter and maybe on Twitter. All right,
Okay,
until next time.
Until next time.